Today we will discuss about role of cell surface glycans towards the towards one of the important cell functionings such as proliferation, migration, and so a figure on the left hand side represents some of the receptors and that has glycans in it. For example, this EGFR receptors has this glycans. Here for the representation only few glycans are shown, but in actually this EGFR has more glycans and those informations are available in this table. For example, this EGFR has 13N glycan site, and what happens when the receptors gets activated through this extracellular matrix, it will trigger this phosphorylation or dimerization. Phosphorylation and dimerization in the cytoplasmic part and cytosolic part, and thereafter it will triggers the signaling pathways such as this MAPK pathways and ERK pathways are triggered by EGFR, and same functions is done by similar kind of functions are done by this FGFR and VGFR also. The difference between these receptors is that both of them activated by all of them activated by different kinds of antigens, and these interactions of these antigens and the receptors also depends on the glycan profiles of the receptors, as this EGFR, FGFR and VGFR has different kind of glycans profiles, and some of these information about the glycans profiles are shown in this table on the right hand side, and also the downstream signaling pathways are might be different or might be same. For example, for this EGFR and FGFR, this signaling pathway that is triggers in the downstream part is almost same as MAPK and ERK pathway. However, for this VGFR, the signaling pathways that are triggered downstream signaling pathways within the cytosols are similar to this EGFR and FGFR, but it includes one more pathway, that is ERK1/2 pathway. And in overall this receptors, that is EGFR, FGFR and VGFR, plays a role towards this proliferation and signaling pathway. That is this activation of this kind of receptors will leads to high proliferations of these cells or high growth rate of these cells, and these things are more prominent in the case of cancerous cells. These things are more activated in the case of cancerous cells. Now let us just quickly go to this MET receptors. MET, that we have also already covered this. What does MET means in many of our previous videos. MET means mesenchymal epithelial transitions. The cells that are in mesenchymal stage are in transitions to or get transited or in transition to or in transitions towards these epithelial characteristics. Mesenchymal means cells can move from one place to another, and epithelial means cell does not move from one place to another. So activation of these receptors will trigger transition of cells from mesenchymal type to epithelial site types, and these receptors, some of these receptors are mentioned here, such as these IGFR receptors, HGF receptors and INSR receptors, and these are also glycosylated receptors, and more informations can be found on the tables in this site, and also for some more informations about these tables can be obtained from the reference that has been provided in the description. So signaling pathways that are triggered in this case also is because of the phosphorylation and dimerization of this IGFR. It will trigger MAPK, P13K, AKTA pathway. MAPK is similar that we can see here. However, along with MAPK, it will also trigger other pathways, and the combined effect of those pathways will cause this MET transition within the cells. And this HGF receptors also helps in this also triggers this MAPK, AKTA, P13K, STAT pathways. And finally, these receptors, this further tyrosine receptors, and all these receptors are tyrosine types receptors. That is, the tyrosine plays a important role in activations of these receptors. Tyrosine is the one of the amino acid that is found in the mammalian cells. Next, this last kind of receptors is this TRKA receptors and RET receptors, and Similar to the previous two categories, they are also glycosylated. That can be shown here. Few glycans are shown here for the representation purpose. Actual information can be found from the table, and it will also trigger MAPK pathway, MAPK AKT pathway. And some of the glycan structures, glycosylation sites are shown in this figure, such as uh, this EGFR. EGFR has this 13 glycan site, and whereas this ERBB2 has 7 glycan sites, and its role and functions are also shown in the last column. That is, EGFR helps in cell migration and proliferation, whereas ERBB2 helps only in proliferations. So this will help us to help you to know how this glycosylation of the receptors plays a role in the important cell functioning, such as migration, proliferation, and these functions are also important when the cells have some tumor characteristics. As one of the common tumor characteristic is to have uncontrolled growth rate, and which is related to cell migration and proliferations. So by targeting this pathway in the tumor cells, we can disrupt the metabolism of the tumor cells, and it will help us to uh, kill or target those tumor cells easily compared to the healthy cells, compared to the traditional ways to targeting tumor cells. I hope you got some basic idea how this cell surface glycans plays a role in this important cell functioning, such as cell migration, proliferation. If you need to understand more about this role of glycans on the cell surface proteins, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.